All right. So everyone, welcome and thank you so much for joining us this evening. We are happy to have Grascen SHIDEMANTLE here with us, Executive Director of the Community Science Institute. But before we welcome Grascen, I just wanted to let you know that tonight's webinar is part of the Lake Friendly Living Awareness Month, hosted by the Lake Friendly Living Coalition of the Finger Lakes. Many of you have attended other programs since May 1st, but just in case this is all new to you, the Lake Friendly Living Coalition includes the following lakes, identified here by this lovely graphic that shows everybody's emblems. So the coalition includes Canandaigua, Cayuga, Canisius, Honeoye, Cayuga, Atisco, Owasco, Seneca, and Skaneateles. And together we work to spread the shared Finger Lakes living message in an effort to improve and protect water quality across the Finger Lakes. Some of you may have joined us in 2021 for the first Lake Friendly Living Awareness Week. And in 2022, we expanded to a month of both in-person and virtual sessions, allowing us to feature this year's theme of watershed resiliency, with a focus on climate change and highlighting components of our Lake Friendly Living programs. If you are interested in learning more about Cayuga Lake's Lake Friendly Living program, please visit our website at cayugalake.org. And I also just wanted to let you know that all the 2022 webinars are recorded and are available now on the network's YouTube channel, the link for which I will put in the chat in just a moment. But without further ado, I am so pleased to introduce tonight's speaker, Grascen SHIDEMANTLE. Grascen is the incoming Executive Director at the Community Science Institute. She recently earned her PhD in biological sciences from Binghamton University, where she researched the effects of aquatic contaminants such as pesticides, road salts, and light pollution on amphibians. Grascen also dedicated much of her scientific career to outreach and education projects, including a National Science Foundation Community Science Project. As the new Executive Director of the Community Science Institute, Grascen is passionate about using scientific knowledge to empower and support the health of her community for the benefit of humans and wildlife. And I know I can join others in the network by saying we are very proud to be in partnership with the Community Science Institute with many programs throughout the last few years. And we are very happy to now partner with Grascen. Grascen, let me turn it over to you. Thank you so much for being here tonight.
Okay, thank you, Jen, for the lovely introduction. I'm just going to pop up my screen here for you all. So as Jen said, my name is Grascen SHIDEMANTLE, and I'm the incoming Executive Director at the Community Science Institute. I'm really excited to be here with you all today to talk about community science and how it's being used to monitor water quality in the Finger Lakes region. A quick note before we get started. You'll notice these captions showing up at the bottom of my screen. This is to help make my talk more accessible to you all, and I really hope that my microphone cooperates and is able to pick up all of the words well for you tonight. The next thing I want you to know is I'll be stopping to ask you all questions during my talk to make this a little bit more of a conversation and less of me talking at you. So when I ask a question or I ask for a contribution, please feel free to type your answer in the chat and I'll read it out loud to share with you all. Okay, so today here's a rundown of what I hope we will be going over. First, we're going to go over a case study of the Community Science Institute's community science success, focusing on the Trumansburg wastewater treatment plant. Next, we're going to define community science and then how it ties into the CSI's mission and our monitoring partnerships. Next, we'll cover some of the benefits and advantages of community science and go over some more specific stories of CSI's community science success. Finally, we'll wrap up the evening with some ways that you all can get involved with community-led water quality monitoring in your region. And then there'll be time for additional questions and discussion at the end. Okay, I'd like to begin tonight with a story. So in 2006, a group of Community Science Institute volunteers known as the Stream Watch Group began monitoring two tributaries of Cayuga Lake, Teganic Creek and Trumansburg Creek. The Trumansburg wastewater treatment plant lays within the Trumansburg Creek watershed. Our Stream Watch group has monitoring sites both upstream and downstream of the plant since 2006. Now, here's my first question where I'm looking for folks to put something in the chat. With a wastewater treatment plant in the watershed, what type of pollutants might you expect to find? And like I said, you can put your answers in the chat and I'll read them out loud to you. Diana suggests E. coli. Great guess. Okay, if anybody has any other suggestions, feel free to put them in the chat, but I am going to move on because Diana predicted correctly. And that's what let's see. All right, let's work on the transitions here. So for several years, the Stream Watch group and CSI staff noticed that E. coli levels, which are an important red flag for pathogenic bacteria contamination, just downstream of the wastewater treatment plant at the outfall, were incredibly high under base flow conditions. On this graph, you see the number of E. coli colonies per 100 milliliters of sample on the Y or the vertical axis, and then the sampling dates all the way back to 2006 on the X or horizontal axis. There's a benefit of community science for you. Look how far back this data set goes. It's beautiful. As you can see on this graph, colony counts numbered in the hundreds of thousands at several time points, exceeding the New York State Department of Health limit for safe swimming. For reference, when New York State parks detect E. coli levels above this level, they're required to close their beaches as they're considered not safe for swimming. Because of these high E. coli levels, CSI met with the village of Trumansburg and the wastewater treatment plant officials several times. However, levels of E. coli still remained high. When E. coli levels again peaked in 2015 and 2016, the CSI Board of Directors wrote to the DEC to report these exceptionally high levels. It was determined that these high E. coli levels were due to aging infrastructure of the plant, and the state obligated the village of Trumansburg to upgrade their plant. Plant upgrades were completed at the end of 2016, and the Stream Watch group has continued to collect water samples to be tested for E. coli and other indicators of wastewater contamination since. And we're happy to say that the plant upgrades worked, and we haven't detected high E. coli levels since the plant upgrades were completed. Now, I'm sharing this story with you all because it's just one example of the ways in which community science can be used to help monitor and therefore protect the health of our local waterways. Oops. Okay, so based on this example I gave you and your experience hearing about community science or citizen science programs, could anyone suggest a definition or some characteristics of community science? You can go ahead and pop those in the chat as well. I used to have to wait on college students to put their answers in the chat, so I can wait all day. Don't be shy. Okay, looks like you picked a good webinar to attend because today you all are going to learn what community science is. So community science is scientific work that's undertaken by volunteer groups from the general public in collaboration with professional scientists. Now, you might sometimes hear the term community science used interchangeably with the term citizen science, and quite often the programs are similar. At CSI, we emphasize that community science differs from citizen science because we work with volunteer groups rather than individual volunteers. Our volunteers work together as a community rather than individually to collect data. So aside from CSI, there are several citizen or community science project examples that you all might be familiar with. One of the most popular is the Audubon Christmas bird count. This one is widespread throughout our area and I believe internationally as well. There's also many iNaturalist citizen or community science programs such as the BioBlitz. And regionally, the Finger Lakes Land Trust has their BioBlitz happening this weekend. So if you're interested in getting involved with that, go to their website. And finally, there is the Globe at Night project, which monitors the night sky for light pollutions. These are just some well-known national programs. Today, we're mostly going to focus on Community Science Institute's community science programs and ways to get involved with monitoring water quality in our region. So as the name suggests, community science is at the heart of what we do at the Community Science Institute, or CSI, as I'm going to be calling it throughout my talk. Our mission is to partner with community-based volunteer groups, aka community scientists, to better understand and protect local streams and lakes by collecting and disseminating scientifically credible regulatory quality data that inform long-term sustainable management strategies. CSI is a 501c3 nonprofit. Most of our funds come from contracts with local county and municipal governments, as well as fee-for-service water testing that we offer for the general public. At CSI, our lab is certified by the New York State Department of Health and the Environmental Protection Agency. This certification ensures that our data are regulatory quality and therefore can be used for management or infrastructure decisions, as we saw in the Trumansburg wastewater treatment plant example. All of the regulatory quality data that our community-based groups collect is uploaded onto our free online water quality database. Data on our database can be easily searched and downloaded, and later tonight, I'm going to be walking you through how you can find data on our database. Next, we monitor water through several volunteer monitoring partnerships, which I'm going to detail for you all in the next slide. And finally, we prioritize outreach and education of water quality as part of our mission. This includes regular speaking events, such as this one today, publication of our annual water bulletin, and a collaboration with the Tompkins County 4-H, known as 4-H2O. At CSI, we work with 250 community-based volunteers in our 4 monitoring partnerships. On this map here, you can see where we have historically monitored water quality in the Finger Lakes region. In our synoptic stream and lake monitoring program, our sampling sites are indicated in blue. Volunteers collect water samples along multiple sites of a tributary on the same day to provide a snapshot of water quality. They then bring these samples back to the lab for certified laboratory analyses. In our red flag monitoring partnerships, represented by red dots here, volunteers collect samples from streams and then perform quality assured measurements themselves in the field. In our biomonitoring partnership, represented by the yellow circles here, volunteers collect samples of benthic macroinvertebrates or stream bugs. And then volunteers sort and identify benthic macroinvertebrate families to determine abundance and diversity, which translates into ecological health of a stream. And finally, in our harmful algal bloom monitoring partnership, volunteers monitor shoreline of Cayuga Lake for harmful algal blooms and collect samples to be analyzed in our certified lab. Today, I would like to focus your attention on our 2 largest monitoring partnerships, our synoptic stream and lake program and our harmful algal bloom program. However, if you have questions about the other 2 programs, I would be happy to discuss them more in detail during the Q&A session. So first, we'll start by reviewing our synoptic stream and lake monitoring partnership. The goal of this program is to produce regulatory quality data on stream and lake water quality that can inform water resource management decisions by local governments. Specifically, we monitor streams and lakes for analytes such as those that are listed here. Collecting this data not only helps us to understand water quality of the streams themselves, but it also allows us to understand how pollutant loading from streams may affect water quality within the lakes. So let me give you a rundown of how this community science project works. Community scientists participate in this program by first attending a volunteer training led by CSI staff. At the training, volunteers are reminded of sampling protocols and safety precautions, and we also share results from last year's sampling with the groups. This training ensures that volunteers are following protocols consistently and safely, and also updates volunteers on the results of their sampling efforts. My personal favorite part of these trainings is that we as scientists get to ask the community members to share their water quality concerns in their monitoring region. This brings us up to date on what's going on throughout our watersheds, and it also allows us to understand trends in our previously collected data. Next, during the training, the volunteer group schedules several base flow sampling events. On the day of the event, volunteers collect samples along several locations of the same stream to get a snapshot of water quality. Volunteers also collect stormwater samples, but for obvious reasons, we're not able to schedule those during the training. We're at the mercy of the weather there. After collecting the samples, volunteers transport their samples to CSI's lab for certified lab analyses. And then once those analyses are performed, the results are uploaded onto our free public online database. So now I just want to brag a little bit about our database, because this is a great reflection of the hard work that our community scientists have put in. The purpose of our online database is to disseminate scientifically credible and regulatory quality results to the public, to local and regional stakeholders, and to government agencies to improve water resource understanding and management. And currently, our database holds over 87,000 regulatory quality measurements of water quality. And to really demonstrate the value of community-led science, I'd like us to take a little detour and visit our database so that you can see the great work that our community scientists have been doing for over 20 years at the Community Science Institute. So I'm going to pop us over to our database. This is the main site for our database. And I'll direct you to Stream and Lakes Chemistry. And the first thing you'll see is a giant map filled with markers. As you zoom in, you can see all of the regions where we and our community scientists have sampled. Blue represents our synoptic stream program. Green represents an investigative sample. So maybe somebody from the public brought a concern to us, so we decided to sample there and see. Red is our red flag program, and yellow is our biological monitoring. Right now, there's no biomonitoring results in our database, but hopefully by the end of the year, that portion of our database will be completed. So the best way to know how to use this is to just explore. So let's say we go to the Cayuga Lake watershed. And I know that today we have several volunteers from the 6 Mile Creek monitoring group. So let's check out water quality on 6 Mile Creek. So when you select your creek, you see a map. You can zoom out, you can zoom in, and each little marker represents a sample site. So when you click on the sample site, it will tell you the name of the site and then some details about where we collect the sample. When you scroll down, you'll see a graph that shows average points for an analyte. The first one that comes up is temperature, but you can look for any parameter that you're interested in understanding. So on the main page for a monitoring set, you'll find all of the different sampling locations and the analyte on the y-axis here. We have both base flow and stormwater data here. And the dots and diamonds here represent the average. So here you can see at German Cross Road, we've taken 32 stormwater samples. There's a benefit of community science. We could never do that on our own. Next, if you're interested in seeing, okay, what's not the average, let's see it in a little bit more detail. We can go to a specific site. You'll see it pop up on the map. And then you can see all of the data we have from the very beginning of sampling. So for this site, it started in 2004. And again, you can change the parameter that you're looking at, and it will be represented on this map. This is really useful because we can see changes and trends over time. So that's just a snapshot of our database. I wanted to show you because it's hard to conceptualize unless you really play around with it. At the end, during our Q&A session, if there's any particular parts of our database that you would like to explore more, I'd be happy to walk through it all with you. Okay, now I'd like to circle back to our monitoring partnerships. So our second largest monitoring partnership is our harmful algal bloom, or HABs monitoring program. Before I describe the program, can someone type in the chat what a HAB is and what might make it so harmful? And I see that earlier when I asked for a definition of a community scientist, it contributed that individuals dedicated to improving the environment were community scientists. And I think that's definitely true, especially of folks who are involved with CSI. Okay, and we have an answer for what is a HAB. And so Darby contributed that a HAB is cyanobacteria that can produce toxins. And that's a great definition of a HAB. A lot of folks think because they're called harmful algal blooms that they're made up of algae, but they're actually made up of cyanobacteria. And the thing that makes them so harmful are those toxins, like Darby mentioned. So it's important to monitor our waterways for HABs because not only are they harmful, but they're also intensifying and increasing over recent years. So in collaboration with the Cayuga Lake Watershed Network and Discover Cayuga Lake, the goal of our harmful algal blooms monitoring program is to collect actionable data on these cyanobacteria blooms so that we can protect public health and relay that bloom information and testing results quickly and efficiently. Whenever our lab receives a HAB sample, we first identify the dominant cyanobacteria genus that makes up the bloom. And then we measure chlorophyll A to understand the bloom biomass, so how dense it is. And then we measure cyanotoxins, such as microcystins, to determine how toxic the bloom is. Like the Synoptic Stream program, the HABs program begins with volunteer training. This involves DEC training on how to identify a bloom, as well as CSI and Cayuga Lake Watershed Network training on how to report and collect a bloom sample. Next, volunteers are assigned a designated segment of shoreline on Cayuga Lake. Every week, that community scientist patrols their shoreline and reports to CSI whether or not there's a bloom. If there is a bloom, the volunteer collects a sample and either they or another volunteer transports it to CSI. At CSI, our scientists perform microscopy to identify the dominant genus of cyanobacteria. They measure chlorophyll A to understand the bloom biomass, and then measure cyanotoxin to determine bloom toxicity. In our lab, we mostly measure microcystin, which is the most common toxin found in HABs, but we've also measured other toxins like anatoxin A. Finally, we report our results to the DEC, as well as upload our data to the HABs reporting page. So now we're going to take another detour to the CSI website so you can see the HABs reporting page. So when you go to the CSI website, if you wanted to find this on your own, if you're on the main page, you go to Monitoring Partnerships, Harmful Algal Bloom, Cayuga Lake HABs reporting page. When you scroll down on this page, you see a beautiful map here. And on this map, you'll see gray portions of shoreline. These portions of shoreline are where our community scientists actively patrol the shoreline to look for HABs. Now, right now, you'll notice it's empty, but there's no HABs marked on the map. And that's a good thing. That means there's no HABs reported so far for 2022. But once we get to the harmful algal bloom season in roughly July through October, this map will begin to be populated with HABs. And you'll be able to read it and see whether or not a bloom is suspicious, whether it contains cyanobacteria, and then if they are confirmed blooms with cyanobacteria, what the levels of microcystin toxins are in them. You can also get data on this page by scrolling down to this master results table, which will show you, excuse me, when the bloom was observed, and additional details. Right now, as I mentioned, there's nothing on here because no blooms have been reported for the 2022 season yet, but tune in later to check out what we would have on there. Also, I'm happy to announce that by the end of 2022, we should have a formal HABs database in the same way that we have our stream and lake monitoring database. So we're very excited for that. Okay, so we've gone over Community Science Institute's monitoring partnerships and the ways that community scientists contribute to those partnerships. And some of the benefits of community science have already started to pop out. Now, there's many benefits. To kind of keep us organized, I've organized them by benefits to scientists, the community, and benefits to both. Can anybody add into the chat some potential benefits of community science? It can be, in general, benefits to society, community members, scientists, anybody. So Ed suggests that a benefit of community science is taking action based on data. And we have another suggestion that education is a benefit of community science. That's so true. Okay, I want one more. Yes, John says that more data means better science knowledge. This is so true. And actually is a great segue to the first benefit that I would like to talk to you about. So first, I'm going to focus on benefits to scientists. So through community science, we're able to collect larger amounts of data across a larger geographical area. So you'll remember those maps that I showed you earlier were huge, and the number of samples we have dating all the way back to the early 2000s, there's so many. CSI staff alone could have never collected all of that data all on our own across such a wide geographical area. Another benefit of community science is that community members are frequently more familiar with their study regions than researchers. So for example, I'm from Western Pennsylvania. I've only lived in New York for 4 years for graduate school. I could never claim to understand the history of some of these watersheds the same way as these people who have grown up in the area, who have seen the landscape change and seen land uses change as well. So by community members contributing that insight to our research and to the data that we collect, we really have a great benefit that we wouldn't have otherwise. Next, let's talk about some benefits to community members. One of the benefits is that community science democratizes science. This simply means that it makes science more accessible to more people. In our community science programs, you don't really need any scientific training to participate. This levels the playing field and means anybody can get involved with science. Next, as somebody mentioned in the chat, community science is a more impactful and effective form of science education, because you're actually learning through doing, not just by sitting in a classroom or in a lecture. And finally, community science in particular helps to build relationship among community members. I mentioned that our volunteers work in volunteer groups, and so they have to coordinate with one another for their stormwater collection, their base flow collection, and this helps to build relationships among them. Now, finally, I'd like to review some benefits to both scientists and community members, and really this goes across the board. The first is that community science helps to build trust in science. And I don't know if any of you have been watching the news lately, but science is having a bit of a PR problem right now, whether it comes to having folks trust climate change science or COVID-19 science. Science is really struggling to get people to trust their results. Through community science, community members are involved with the process. So there's no secrecy involved. Everybody knows how it works and what's happening. And finally, my favorite benefit for community science is that community members can directly participate in the regulatory framework to protect and manage shared natural resources. And this goes along with what somebody had mentioned in the chat and is a real strong suit, I feel, of the Community Science Institute in particular. So for the end of today, I want to share a couple more community science success stories from CSI. This next community success story, this next community science success story is one that ties in two of CSI's partners. So in 2013 and 2018, respectively, the Seneca Lake Pure Waters Association and the Cayuga Lake Association began collaborating with CSI. And I think we have some representatives from those two groups here today. So hello to you both. These SLPWA and KLA organize groups of volunteers like we do in the Cayuga Lake watershed, and then they transport the samples to our certified lab, where we analyze them for many of the same parameters that we measure in our synoptic stream program within the Cayuga Lake watershed. Currently, these data sets, which includes nearly 10 years of data on Seneca Lake, are being used as the foundation for a model of the Seneca-Cayuga Lake 9-element watershed management plan, or the 9E plan. This type of watershed management plan is a lot less costly and time-consuming than your typical total maximum daily load. In my brief time here at CSI, I have to say that this project has been one of the most inspiring to me because it emphasizes what we can accomplish when we work together for this common goal of high quality science for the sake of high quality water. I've been really inspired by the work that the folks at SLPWA and KLA have done in collaboration with us and their groups of volunteers. And I did just want to share a fun fact with you all that I read that emphasizes one of the reasons why protecting Seneca Lake and Cayuga Lake water quality is so important. And it's that these two lakes make up a significant portion of the water in the Finger Lakes. Did you all know that Seneca Lake and Cayuga Lake make up 50% of the water in the Finger Lakes? That blew my mind when I found that out. So just another point in why we need to protect those waterways. Now, if you want to learn more about the Seneca-Cayuga 9E plan, you can read our water bulletin, fall 2021. I'm going to take this link and pop it in the chat for you all so that you can read that later if you'd like. All right, there's the link for you. And there's lots of other good stuff to read in there too. You can also read about how we have helped to monitor E. coli in the Canandaigua Lake watershed as well. Okay, so I'd like to share just one more community science success story with you all tonight. Until 2014, the southern shelf of Cayuga Lake was listed on the DEC's 303D list of impaired water bodies for high levels of phosphorus and E. coli. With data collected during our monitoring cruises, CSI was able to successfully delist the southern shelf as impaired by pathogenic bacteria or E. coli. On this graph, we have the number of E. coli colonies per 100 milliliter sample on the Y or the vertical axis. And then on the X or the horizontal axis here, we have sampling locations along within Cayuga Lake, excuse me. The blue dots represent the average E. coli over all monitoring events. And the red bar represents the Department of Health's contact recreation limit for E. coli. This is the same limit that we discussed when we were talking about the wastewater treatment plant at the beginning. As you can see, all of these data points fall well below the contact recreation limit. This data, largely collected by young community scientists during our 4H2O water quality cruises, played a crucial role in removing the southern portion of Cayuga Lake from the 303D list in regards to pathogenic bacteria. This delisting is important because it shows that Cayuga Lake is a high quality resource for drinking water, swimming, and recreation, which is essential for both public and environmental health, as well as the health of businesses. Okay, so for tonight, I would like to wrap up by talking about some ways that you can get involved in water quality monitoring as a community scientist in the Finger Lakes region. First, I'll talk about how you can get involved with CSI, how you can get involved with CSI's partners, and then how you can also get involved with some DEC statewide programs. So first we'll start with CSI. So citizen scientists can get involved with our synoptic stream and lake monitoring program. This largely happens in the Cayuga Lake watershed, but on the next slide, I'll talk to you about how you can get involved with this type of monitoring with our partners on Seneca, Cayuga, and Canandaigua Lake. We're also currently recruiting volunteers for our harmful algal bloom monitoring season. We call those community scientists HABS Harriers. If you're interested in volunteering for that, please send us an email. I'll show you the email at the end of my talk. We're also always recruiting folks for our biomonitoring and red flag monitoring program. Currently, our biomonitoring program has historically sampled in the Cayuga Lake, Seneca Lake, Owasco Lake, and Susquehanna River watersheds, but we're always looking to expand. So if you would like to get into biomonitoring with us, please reach out. And our red flag monitoring can happen really anywhere in the state, as long as you don't mind making a trip to Ithaca every now and again to bring us a quality assurance sample. Now, say you're not in the Cayuga Lake watershed area, but you're in the Canandaigua Lake watershed, Cayuga Lake watershed, or Seneca Lake watershed. We do have monitoring partnerships with these three groups. These three groups organize the volunteers and then bring the samples to our lab. So you're doing similar sampling to that which we do at CSI. The only difference is that these groups organize the volunteers. We just measure the samples. And finally, there are two regional or statewide monitoring programs, which you might be interested in learning more about. The first is the Citizens Statewide Lake Assessment Program, or CSLAP. In this program, community scientists monitor water quality of lakes. And this is led by the DEC and the New York State Federation of Lake Associations. Next, there's the Water Assessment by Volunteer Evaluators, or WAVE program. And this is a stream biomonitoring program that monitors for benthic macroinvertebrates, similar to our biomonitoring program. This is again a New York State DEC program, and it's largely used to determine where further water quality monitoring, specifically water chemistry, should be done through the DEC's rotating integrated basin program. So 3 main ways to get involved: through CSI, through our partners, or through these two DEC citizen science projects. And that is all I had prepared for you all today. I would just like to say a big thank you to the Lake Friendly Living Coalition, and especially to Liz and Jen at the Cayuga Lake Watershed Network for hosting us tonight. It was so wonderful to be here with you all. If you would like to volunteer and to be a community scientist with the Community Science Institute, please email us at info@communityscience.org, and we will help you get all acclimated into our program. If you have specific questions for me, feel free to email me at my email address here. This is our website if you would like to learn more about what we do, and you can also keep up with us on Facebook and Instagram. And with that, if anybody has any questions, I'd be happy to take them now or hear any of your feedback.
Thank you so much, Grascen.
Of course.
That was fantastic. Really, really appreciate that. Grascen mentioned, I know several folks on this presentation tonight are already kind of dialed into CSI, maybe are doing some HABS monitoring, part of some of the other programs that Grascen mentioned this evening. But if there are any final questions or anything else anyone would like to know, now is the time to add your questions to the chat. Feel free.
Thank you, everybody, for your kind words.
That's fantastic. We'll give it just another couple seconds. Grascen, it was great for you to include folks along the way. As you went through the program.
Thank you all for humoring me on that.
Question from John Abel.
Yeah, okay, so John asked, please tell us more about how you ended up in this position. So for me, I can tell you my personal journey to this position was that I was always motivated by my love of the environment and of wildlife. And the most important thing to me was always to do something that made a difference, but also gave back to the community. So I went to graduate school to understand how pollutants impact wildlife. And I felt like that research was important, but it didn't have the community impact that I was looking for. So when I graduated a couple weeks ago, before that, I was looking for a position where I could really integrate scientific integrity with community service. And so as a graduate student in New York, I fell in love with the area, and you guys can't get rid of me now. I love New York State so much. And so when the position at CSI popped up, I just thought this is exactly what I've been looking for, something where I can integrate my scientific knowledge with my deep passion for community engagement. So that's how I personally ended up here.
Thanks, Grascen. Thanks for inviting that elaboration. I'm sure many of us were curious.
Yes, I started as a biologist. And so if anybody, I'm always here to learn. I'm learning more and more about water quality every day. I understood it in an ecological context, but now I'm learning about it more in a public health context and in terms of water chemistry. So it's been an interesting past few months getting up to speed.
Only imagine, and you're just such a busy time of the year here with the work that you're doing.
Well, just like I said, the best way to learn is to do it. So that's what we're doing.
That's great. Folks, any other questions? I'm not seeing anything else in the chat. Grascen, I thank you for providing your contact information. I'm sure you'll be from folks after the fact. This presentation, like all the rest, will be uploaded to our YouTube channel, the network's YouTube channel, and we will let all of you know when that's available, so you can go back and look at any of the research that Grascen mentioned, look at some of the websites that she mentioned. Definitely check out their data sets on their website, sign up for some monitoring, perhaps some other volunteer opportunities that are available, and share this information with others. Yeah, Grascen, I think we're set for this evening.
Yeah, great.
All right. Thank you so much. Thank you very much. And thanks everyone for attending, and have a lovely evening. Thanks. Bye.